Microscope. Microscope is an optical device which makes an image of a small object to appear very large. When the size of the image is larger than the size of the object, the image is said to be magnified. In other words, the image is said to be magnified when the ratio of the size of the image is larger than the size of the object. The ratio of the size of the image represented as V to the size of the object represented as U is called magnification factor. That is, V divided by U is equal to M. Similarly, if the size of the image is smaller than the size of the object, then the image is said to be diminished. That is, for V divided by U is equal to M. If M is greater than 1, then the image is said to be magnified. If M is lesser than 1, then the image is said to be diminished. Now, let's see convex lens as a magnifier. Write the word physics on a white sheet of paper. Now, take a convex lens and adjust the distance between the word on the paper and the lens suitably so that the letters of the word physics appear big. Hence, a convex lens is also called a simple microscope. Principle and working of a compound microscope. To obtain greater magnification, an optical device called compound microscope is used. A compound microscope is a combination of two convex lenses whose focal lengths are different. The lens with the short focal length f will be facing the object. This lens is called objective. The lens with large focal length capital F is used for viewing the object, and this lens is called eyepiece. The two lenses are so arranged that their optic centers lie in the same line. The objective and the eyepiece are fitted to one end of the two separate tubes. The two tubes are such that one tube fits into the other and slides freely. The distance d between the objective and the eyepiece can be adjusted by sliding the objective, holding the eyepiece fixed. Working principle. When a small object AB is placed beyond the principal focus of the objective O, a magnified image, real and inverted I1G1 is formed on the other side of the objective beyond to F. Now, this image I1G1 serves as an object to the eyepiece E. The distance between this image and the eyepiece is adjusted so that the image falls within the principal focus of the eyepiece. The second and the final image, I2G2, is greatly magnified, virtual and inverted. Seeing an inverted image is not a disadvantage, as the microscope is used to observe very small objects such as bacteria, blood cells, to measure the diameter of a capillary or cross-section of a thin wire, etc.